Hello everyone. Today I'm going to give you the brief idea about the endocytosis, pinocytosis and phagocytosis. These are the some import export transport system in the cell which play important role. So for proper functioning of the cell needs to import things from outside to inside the cell, and for its proper functioning they need to export some components or some products from inside to the outside. When things are coming from outside to inside the cell, this is called the import. It includes we have to import oxygen, sugars, amino acid, and even small ions. For export, this after the metabolic activity there will be the metabolic waste within the cell. It should be excreted out. The carbon dioxide we have to expel out, and some secretory proteins which are formed on the endoplasmic reticulum. Then from the Golgi bodies they make the vesicles. This protein has been sent outside the cell. The process of movement of large molecules from inside the cell to the outside the cell is called as exocytosis. All the processes, either endocytosis or the exocytosis, is carried by the vesicle formation. As you can see in the diagram, we are having extracyte fluid, and there is one process called phagocytosis. When the molecule are large sized, like the bacteria, so these our cells engulf these bacteria within in the membrane. Then these membrane bound vesicle will go and get merge with the lysosomes, which are having hydrolytic enzyme. Then the breakdown of this bacteria which is coming by the phagocytosis process will get breakdown. So on the product categories we are having endocytosis and exocytosis. Endocytosis means when material are coming from outside inside the cell. This is the endocytosis, and simply exocytosis is the material going from cell to the outside. So in endocytosis we are having phagocytosis, or simply endocytosis, or pinocytosis. Like here, first I said, in phagocytosis, if there is the larger molecule, or this is the bacteria is larger in size, so this bacteria is engulfed by the cell and form the phagosome along with the lysosome, or we can say that when bacteria is present on the surface of the plasma membrane of the cell, it get engulfed then in the vesicular form. Then this vesicle will go and bind with the lysosome, where this phagocytic cell is converted into the phagosomes, where phagosomes along with the lysosomes are interacting. Then there is a breakdown of this bacteria or the larger molecule. We have studied this in the lysosomes. Now the another is the fluid phase of endocytosis. What is happening? Whatever the material is outside the cell, they are simply coming in the form of vesicle without any specification. Every particle, whatever is present outside the cell, they are coming into the cell in the form of vesicles. And in another type, which is called as also pinocytosis, here receptor mediated endocytosis takes place. We are having specific receptors on the surface which selectively absorb material from outside the cell. Then these are taken in the vesicular form. Then the process is carried out. We will see this detail in the later. Then Other is the exocytosis process. In case of exocytosis process, whatever the waste product has to be sent outside the cell, they will be in the form of vesicle. Then these vesicles will join the plasma membrane and throw its product outside or throw its product into the bloodstream. So these are the three main basic processes which are involved in the import and export of material inside the cell. So the endocytosis term was first given by Christian Didouy in 1963. The term endocytosis was first coined by Christian Didouy in 1963, where he said the ingestion of larger molecules or intake of larger molecules such as bacteria is the one process of endocytosis, and the uptake of fluid or macromolecules in the form of vesicle by the cell is the endocytosis process. In endocytosis process, material are internalized within the membrane. Then this membrane is budding off. From the plasma membrane. Then they come inside the cell. Then, as per their location, they will go and fuse with the lysosomes, and the process will be carried out. We will study this in detail. So we can say that endocytosis is the simply internalization of material which is surrounded by the membrane. This membrane buds off from the plasma membrane, forms the vesicle. Then this vesicle go and bind with the lysosomes, where degradation process occurs. Now we are having receptor mediated endocytosis. In this receptor mediated endocytosis is also called as pinocytosis, in which there is selection of material or selectively uptake of material. Macromolecules are taken which are internalized. First, they have to bind to the receptors which are present on the plasma membrane. So there is the selection of the macromolecules by the receptors which are present on the plasma membrane. So here I have given you one example, that is the Catherine coated pits. So what is happening? This is the Catherine coated endocytosis or receptor mediated endocytosis. We are having receptors on the plasma membrane, but when the molecules are getting attached, like the molecules getting protein or any other component getting attached on the surface of the receptors, they are there, but we have to form this in the vesicle form. So these Catherine which are present in the cytosol go and attach these receptor molecule. So they pull these receptor molecules, they allow plasma membrane to get bent in the form of bud. Like here, they form the bud. Then this bud is pinched off from the plasma membrane. They form the vesicle. Then this vesicle will go and form the early endosome, where they will deliver the material whatever they have taken from the outside cell. Then this early endosome will mix up with the lysosome. They will form the late endosome. Then the hydrolysis process takes place. But we are having one peculiarity things here. The receptor and the Catherine which are in the vesicle are recycled back. They are recycled back, going back to the plasma membrane, where they will again follow the same process. This process is associated with the GTP binding protein. This is associated with the GTP binding protein. It means this is a energy consuming process. So this is how receptor mediated endocytosis takes place. So in this diagram, I have given you the example of the LDL. So what is this LDL? This is a low density lipoprotein. This is the low density lipoprotein. As we know, cholesterol is transported in the bloodstream in the form of lipoprotein particles. कोलेस्ट्रॉल जो हमारी ब्लड में होता है वो किस फॉर्म में होता है? लिपोप्रोटीन पार्टिकल्स में होता है. उसको हम लोग कॉमनली कहते हैं LDL, लो डेंसिटी लिपोप्रोटीन पार्टिकल्स. व्हेन दे आर इन द ब्लड स्ट्रीम, we are having specific receptors for this LDL. This LDL will get bind with the specific receptors which are present on the plasma membrane. Then, due to the presence of cathrin at the base, there is a depression in the plasma membrane. Due to this depression, the cathrin proteins make it to form a bud-like structure. Then the LDL will come into this bud. I have missed this thing. The LDL will come into the bud and form a complete vesicle. Then the protein when they are inside the cell, they will go and join with the lysosomes, where different small molecules are formed, like amino acids or some other molecules are formed inside the cell. But the remaining part, which is the catherins are there, the receptors are there, they are sent back to the plasma membrane, where they will again take the another molecule of cholesterol inside the cell for the breakdown. So here we are having another simply the phagocytosis. What is happening in phagocytosis? We are having bacteria cell. So bacteria cell is engulfed within the membrane of the plasma membrane. Then they form the phagocytosis. Then phagosomes, we can call these. By the process of phagocytosis, the bacteria within in the phagosome are called as phagosomes. When these phagosomes get mixed up, interact with the lysosome, they form the phagolysosomes. Then the whole breakage of material takes place. There was a scientist, Michael Brown and Joseph Goldstein. There were two scientists who worked in the laboratory, Michael Brown and Joseph Goldstein. They only demonstrated that the LDL in mammalian cell require specific surface receptors. These are concentrated with the cathrin coated pits, and then this cholesterol is internalized by the process of endocytosis. This was the brief concept about the pinocytosis or receptor mediated endocytosis. Now the last we have to give brief about the phagocytosis. We have discussed this phagocytosis in the earlier lectures as well. It is the simple ingestion of the or intake of the larger molecules or microorganisms by the cell, which form the phagosomes. Then these phagosomes are taken by the lysosomes, which are forming the phagolysosome. Then the digestion process occurs. This is the one of the important function of the phagocytic cells or phagosomes, in which if there is a bacteria, by this process we are able to eradicate bacteria from our body, or we remove invading bacteria. These phagocytic cells are having size of 1 to 2 micrometer in diameter. 1 to 2 micrometer in diameter, which is almost the same size of the phagocytic cells. And this phagocytosis always occur on extracellular surface, क्योंकि phagocytosis कहाँ बाहर से आना चाहिए. So it always occur on the extracellular surface. In phagosomes, they get mixed up with the acid hydrolases which are present in the lysosomes. As we know, lysosomes are having many different acid hydrolases which are responsible for the breakdown of the macromolecules. Like we are having nucleases which are responsible for the breakdown of the nucleic acid. We are having proteases which are responsible for the breakdown of the proteins into the amino acids, and similarly we are having lipases and so many different enzymes. So the phagocytosis is having distinct role in different cells. If we take example of amoeba, they use this phagocytosis to capture the food. So this phagocytosis in other organisms are having distinct function. Like in case of amoeba, protozoans or bacteria, they use phagocytosis process to capture their food. But in case of multicellular animals, this phagocytosis act as it provides a defense mechanism, as it eliminates aged or damaged cell from the body. So here we complete our brief idea about the endocytosis, receptor mediated endocytosis, and the phagocytosis. Thank you.